Imagine you are in the microbiology lab. You look at a gram stain, and you see purple rods. Now the question is, what are they? Are they harmless normal flora? Are they a foodborne organism? Or could they be something serious, like Bacillus anthracis? This is why gram-positive bacilli can feel confusing, but we can make it simple. Instead of memorizing every organism alone, we will follow a few clues: spores, motility, hemolysis, catalase, acid-fast staining, and clinical context. By the end, you will have a simple pathway to identify the most important gram-positive bacilli. First, let's organize the group. Gram-positive bacilli are not all the same. Some form spores, like Bacillus. Some do not form spores, like Listeria and Corynebacterium. Some look pleomorphic, meaning their shapes are irregular. And some are branching or filamentous, like Nocardia. So before asking, what organism is this? Ask a simpler question: does it form spores? Is it branching? Is it modified acid-fast positive? This first step makes the whole topic much easier. Now let's start with the spore formers. The first organism is Bacillus anthracis. This one matters because it is a BSL-3 pathogen. That means the lab must handle it very carefully. Clinically, it can cause cutaneous, inhalational, or gastrointestinal anthrax. At the bench, look for this pattern: large rectangular gram-positive rods, spores, catalase positive, non-motile, and non-hemolytic. The classic colony clue is the medusa head appearance. A simple way to remember it is: Bacillus anthracis does not move, does not hemolyze, and forms medusa head colonies. That pattern should immediately raise concern. Now let's compare Bacillus anthracis with Bacillus cereus. This comparison is very high yield. Bacillus anthracis is non-motile, non-hemolytic, and tenacious. It is linked to anthrax. Bacillus cereus is different. It is motile, beta-hemolytic, and non-tenacious. It is linked to foodborne illness, especially rice dishes that were not stored properly. So remember this simple contrast. Anthracis: no movement, no hemolysis. Cereus: moves and beta-hemolyzes. That one comparison can save you a lot of confusion. Now let's move to Listeria monocytogenes. Listeria is a short gram-positive rod. It does not form spores. One important feature is that it can grow at 4 degrees Celsius. That means refrigeration does not fully stop it. So contaminated refrigerated food can still be risky. Clinically, Listeria is especially important in pregnant women, newborns, elderly patients, and immunocompromised patients. So when you hear Listeria, think refrigerated food plus high-risk patients. Now let's see how the lab identifies it. Listeria has a few classic lab clues. It is catalase positive, it is esculin positive, and it has a very special movement pattern. At room temperature, it shows tumbling motility. In semisolid agar, this creates an umbrella pattern. On blood agar, Listeria can show a narrow zone of beta-hemolysis. The CAMP test can also help. It produces a block-like or rectangular zone of enhanced hemolysis. So the simple Listeria pattern is tumbling motility, umbrella growth, narrow beta-hemolysis, and positive CAMP reaction. That is the story of Listeria at the bench. Next is Corynebacterium diphtheriae. This organism has a very recognizable shape. It appears as pleomorphic, club-shaped gram-positive rods. They may line up in a palisade pattern. You can think of them as irregular rods arranged at angles. Clinically, this organism causes diphtheria. The key clinical clue is a gray or white pseudomembrane in the throat. In the lab, special tellurite media can show brown-black colonies. But here is the most important point. Shape alone is not enough. For Corynebacterium diphtheriae, the big question is, does it produce toxin? That must be confirmed by ELEC test, ELISA, or PCR. Now let's separate a few Corynebacterium species. Corynebacterium diphtheriae is linked to diphtheria and pseudomembrane. It is urease negative. Corynebacterium ureolyticum is linked to urinary tract infections. The key clue is rapid urease positivity. Corynebacterium jacquium is linked to catheter lines, prosthetic devices, and endocarditis. It may also be multi-drug resistant. Here is the practical point. Sometimes Corynebacterium looks like normal skin flora, but context matters. If the patient has a catheter, prosthetic device, or urinary infection, do not ignore it too quickly. Now we move to a very recognizable organism, Nocardia. Nocardia looks like long, thin, branching gram-positive rods. The most important clue is modified acid-fast positive. So if you see branching gram-positive rods, and they are modified acid-fast positive, think Nocardia. Its colonies may start chalky white, and later become salmon orange or pink. Clinically, it can cause soft tissue infection. It can also cause pulmonary or disseminated disease, sometimes involving the central nervous system. The memory clue is simple. Branching plus modified acid-fast equals Nocardia. Now let's simplify 2 organisms with special clues. First, Erysipelothrix rhizopathii. It can cause erysipeloid cellulitis. It is associated with animal exposure, such as butchers, anglers, and farmers. At the bench, remember, Catalase negative, non-motile, and H2S positive on TSI. That H2S causes blackening in the tube, so for Erysipelothrix, remember, H2S on TSI. Now Arcanobacterium hemolyticum. It can cause pharyngitis in older children. It is beta hemolytic and catalase negative. The key clue is the reverse camp test, so keep it simple. Erysipelothrix equals H2S. Arcanobacterium equals reverse camp. Now we have two more pattern recognition organisms. First, Rhodococcus equi. It is important in immunocompromised patients. It may cause bloodstream infection, pneumonia, or skin disease. It can be modified acid-fast positive, similar to Nocardia. But the colony clue is helpful. Salmon-colored and slimy colonies. Now let's look at Tropheryma whipplei. This organism causes Whipple disease. Unlike many organisms in this video, it is not usually diagnosed by routine culture. Diagnosis depends more on tissue biopsy or molecular methods. The key stain pattern is PAS positive, acid-fast negative. So remember, Rhodococcus, modified acid-fast positive. Tropheryma, PAS positive, acid-fast negative. This slide shows an important idea. Microbiology is not always about finding one dangerous pathogen. Sometimes disease happens when normal flora changes. On the left we have Lactobacilli. They are protective normal flora in the mouth, GI tract, and female genital tract. They help protect against infection. On the right we have Gardnerella vaginalis. It is associated with bacterial vaginosis. A key clue is the loss of protective lactobacilli. Another clue is the presence of clue cells. These are epithelial cells covered with bacteria. So the big idea is, sometimes disease comes from an imbalance, not just from one organism appearing. Now let's bring everything together. This final matrix is your study tool. Do not memorize these organisms randomly. Use a pattern. Ask, is it catalase positive? Is it motile? Does it form spores? Is there hemolysis? Is it acid-fast? What is the clinical context? Let's review the big clues. Bacillus anthracis, non-motile, non-hemolytic, medusa head colonies. Bacillus cereus, motile, beta hemolytic, foodborne illness. Listeria, tumbling motility, umbrella growth, narrow beta hemolysis. Erysipelothrix, H2S positive on TSI. Arcanobacterium, reverse camp test. Corynebacterium, diphtheriae, brown-black colonies, and toxin conformation. Nocardia, branching and modified acid-fast positive. So when you see gram-positive rods, slow down. Do not guess. Follow the clues. Spores, motility, hemolysis, catalase, acid-fast staining, and clinical context. That turns gram-positive bacilli from a memorization list into a logical identification pathway. If this helped you, subscribe to Lab Science Simplified for more CLS and MLT microbiology reviews.